All right, we're going to get started. If everyone wants to take a seat, grab your last thing of caffeine and sugar. And while you are getting situated, there are these surveys on the table. So if you wouldn't mind just taking a few minutes, like probably just 1 minute even, and filling it out. And you can just leave your completed one on the table and we'll come by and we'll pick those up. But that really helps us for next year's factor, right? Keep making it better. So thank you for doing that. And we have our last panel of the conference, you guys. We made it. We persevered. And what a panel, right? Like, what a great panel. So I am pleased to introduce Andrew, who will introduce our next moderator.
Do the paper. Did you take? Okay, there we go. Hello, my name is Andrew. You may have remembered me from the panel just a short little bit ago. But just as a reminder, I was diagnosed in 2017 in my right proximal femur. I went through the standard MAP treatment, got a limb salvage surgery, but I've been coming up on 6 or 7 years NED so far. Thank you. Dr. Katie Albert is an associate professor at the University of Washington School of Medicine and an attending physician at Seattle Children's Hospital, where she serves as the medical director of the solid tumor team and co-leads the multidisciplinary sarcoma program. She received her undergraduate degree from Brandeis University and her medical degree from the George Washington University School of Medicine. She completed her residency training at Johns Hopkins University and her fellowship in pediatric hematology oncology through the Johns Hopkins University National Cancer Institute Combined Program. While a fellow and junior faculty at Johns Hopkins, she worked in the lab of David Loeb, studying molecular targets of pediatric sarcoma with a focus on rhabdomyosarcoma. Since joining the faculty at Seattle Children's, her research focus has been on the development and conduct of immunotherapy clinical trials for high-risk pediatric and young adult solid tumors. Please help me welcome Dr. Katie Albert.
Great, thanks everybody. You made it to the best panel ever. It's great to see so many people still here at the end of the conference, and I think we have a really great and interesting panel, complementary panel for you guys that will be really of interest to our scientists, our clinicians, patients, and families. So I'm really excited to introduce this panel. First, just to say thank you to Christina for her organization and MIB for putting on another really fantastic conference that I think all of us really look forward to attending every year. And also thank you, I am on a travel, part of my travel was awarded from the family of Victoria Marsh. And as I was reading and learning about Victoria, who is now an osteoangel, but was diagnosed about 10 years ago and had metastatic osteosarcoma at diagnosis, and similar to many of the patients that we know and treat as clinicians, put her life on hold to do upfront therapy. And was in 7th grade at the time and a competitive swimmer and very social, active in school and with her friends. And at the end of therapy, sort of reintegrated and was excited to kind of get back to life, but unfortunately continued to have metastatic recurrences. And also, like many of the patients that we know and those of us who do clinical trials especially get to meet, had to begin living life with osteosarcoma, and unlike with upfront therapy, went back to school, graduated from high school, continued in schooling, got back to swimming, planned a lot of fun trips with her family and friends. And it just kind of reminds me of so many of the patients that we meet and talk to about the trials we're doing, and take to heart really those like individual stories and, personalized goals that patients bring with them, especially in these high-risk situations. So really grateful for that, and definitely thinking about the memories of our patients. So we've heard a lot from our scientists and investigators who are trying to figure out the biology of osteosarcoma and what we can bring forward to patients, and so much good work being done. Finally, we get to talk a little bit about what happens when we are actually ready to bring therapies to the clinic and introduce them to patients. And this panel is going to explore both some specific clinical trials and provide some new and exciting data to think about, but also thinking more generally about how do we practically and thoughtfully bring trials forward to get the highest impact to both the participants on the trials and our future patients, so that when we complete these trials, we can feel really good about the data we've collected and that it will really thrust us forward in terms of finding better therapies. So I'll just now briefly introduce our panel, and then we can get started with the talks. So our first presentation is going to be from Dr. Alanna Church from Dana-Farber, who's a pediatric pathologist, who will talk to us about the clinical implementation of the vast data we're learning about the genetics and molecular characteristics of osteosarcoma. Then we'll hear a talk from Dr. Brenda Weigel, who's from St. Jude Children's Research Hospital and expert extraordinaire on clinical trials, just speaking more generally about how we make collaborations with other institutions, with the federal government, with pharma and industry, to actually bring these trials and these concepts to fruition. And then we'll go into some talks about specific clinical trials. So Dr. Damon Reed from Memorial Sloan Kettering is going to present data from a phase 2 trial. I won't call it an immunotherapy, a HER2-directed therapy for relapsed osteosarcoma patients who have achieved remission. Then Dr. Kristen VanHeyst of Rainbow Babies and Children's Hospital will present on targeting TGF-beta signaling in the microenvironment for osteosarcoma. And then finally, Dr. John Ligon from the University of Florida will discuss utilizing RNA lipid particles that are targeted for osteosarcoma in addition to other solid tumors. So thanks for sticking around. I think this is going to be really great, and we'll go ahead and get started with Dr. Church.
Thank you so much. So thanks all so much for being here with us at the very end of the conference. I'm really grateful to be here with you and with this amazing panel. I appreciate all of you for adopting a pathologist into your group. I also want to thank Buddy and his family for providing a travel award so I could be here with you today. Let me see if I know how to do this. Yes. So I've been in Boston now for a while, but I'm Canadian, and I'm showing you a photo of one of our Canadian heroes, Terry Fox. Terry was diagnosed with osteosarcoma in 1978. He had an amputation, and he was treated with methotrexate and adriamycin. Soon after his treatment, he began a marathon of hope where he aimed to run across Canada to raise money to help others with cancer. He died in 1981 from his osteosarcoma, and his family continued his legacy with a foundation that organizes the Terry Fox Run every year. So like all Canadian children, I participated in this run every year. We knocked on all our neighbors' doors to ask for donations, and we ran around the track, even though I have very limited athletic ability. And their foundation has now raised over $900 million Canadian dollars for cancer research. So he and his family are really inspiring, like all of you here today. So there have been a lot of improvements since Terry's time. We can see that I think our prostheses are better, but the chemotherapy that he got is really similar to what we're still giving patients today, and we know we can do better. So one of the many challenges that we face in cancer care is moving all of the discoveries, like many of the advances that we've heard about here this week, including molecular profiling and new targeted treatments, from research into everyday clinical practice. And it can take years before all of these advances reach our patients, and even longer to become really available to all of our patients. So some of the barriers that we face include lack of funding for clinical trials, regulatory approval, and insurance coverage. As a result, not every child or adult has equal access to the most advanced testing or treatment. And bridging this gap is really essential to making the best care available to everyone. So as we think about implementation science, or how to be effective with clinical implementation, we need to look at the whole picture of what's happening, what's needed. There are a lot of steps in launching any new initiative, whether it be a test or a treatment, and in optimizing and improving it over time. So today I'll share with you some of the work that we've been doing in implementing precision medicine and molecular genetic profiling, since this is my area, but implementation science can be applied to any area of clinical medicine. To have a complete cycle of testing and treatment for patient care, we need the whole workflow, ordering, testing, reporting, treatment delivery, and billing to all be robustly established within the clinical environment. And this includes CLIA validation of every test that gets returned into the medical record. Implementation is a difficult area to work in because it's really unglamorous, it's difficult to get funded, and we're often facing barriers that feel really frustrating, like why won't an insurance company pay for this test or treatment when we have so much evidence that it can be helpful? But we persevere, and we have to really be relentless in this area to address every obstacle that comes up. So we approach this work by asking, what can we do today to make sure that a child diagnosed with osteosarcoma or another cancer a couple years from now in a small community far from a pediatric cancer center gets the best care, and that includes molecular profiling to understand and act on the genetic profile of the cancer. So some of the challenges that we face include ordering, billing and payment, and we also deal with a lot of regulation in the laboratory space. So there are a variety of clinical ordering practices. We heard today from the patient panel about some of the variability that they encountered in treatments that were offered, discussion of fertility. There's also differences in how people order tests at places across the country. We really want to get the right test at the right time, ideally early in the course of therapy. There are some guidelines, including NCCN guidelines about testing for certain diseases, but there are a lot of gaps. I'm showing some of the work from myself and one of my colleagues, Catherine Gestrich, who's at the University of Pittsburgh. We did a survey of our pediatric pathologists across the country and asked them how they're ordering molecular tests and how it's going for them, how comfortable do they feel with it. What we can see here is that they are convinced that molecular profiling is useful. They do have some discomfort with some aspects of it, and the ones that they called out specifically were really around billing and reimbursement. There was concern they didn't want to saddle a family with another bill when they're going through a difficult time at the time of diagnosis. We also asked them about what helps them, and we showed that their comfort with the technical details and the clinical impact of the testing that they're ordering really correlates with having a trusted colleague who helps them in supporting the process. So these are some of the things that we have to think about as we're trying to build a robust system. There's a lot of complexity to genetic testing. We have been fortunate to work with patients and participants, particularly through the Count Me In study and the participant engagement in cancer genomic sequencing study through the NCI Cancer Moonshot, and we just published a new paper with I highlighted our two MIB friends here who helped us help inform our thinking. And we're advocating here for having a more integrated approach to how we do our genetic testing for patients with cancer. So in the current model, we have germline diagnostics, where we think about identifying a genetic alteration that may inform the patient's risk or familial inherited risk of cancer, and then the tumor profiling that we do, and they're pretty separate. So we're trying to advocate for a more integrated process that can help, help our patients understand what they need to do next, and also to support our clinicians in that process. There's a ton of complexity in the billing environment. I have like a shallow understanding of this, but there's a lot of coding that needs to be done every time we bring a new molecular test into the lab, and we're lucky that we have a great team that helps us work on this. One of the things that we're trying to do to address many of the barriers that we're facing with respect to billing is to create some guidance for pathologists and oncologists to know what tests to order when, and really to compel our payers to cover the costs of these tests. So there are guidelines out there for certain pediatric cancer diagnoses, but there are a lot of gaps, mostly because pediatric cancer really is a collection of many different rare and ultra-rare diagnoses. So we're aiming to write guidelines that will be the first to address molecular testing for all children with cancer. This is a working group that's in progress. We just met this week, so we have these draft guidelines that I'll share with you. We reviewed the literature about the clinical utility of molecular diagnostics, and we have good evidence from a lot of studies that have been published, and some by people in this room, that molecular profiling using next-generation sequencing, methylation profiling can really improve our diagnostic yields. They can improve risk stratification and identify opportunities for match-targeted therapy. So these are our draft guidelines, but we're making a recommendation that at the time of diagnosis, every child, adolescent, and young adult with a new diagnosis of a solid tumor, and we include brain tumors in here as well, receive DNA-based next-generation sequencing, that everyone receives RNA-based sequencing to detect fusions, and that children with brain tumors also get methylation-based tumor classification. We're recommending that at the time of relapse, if they haven't already had those other tests, that we go back and do them. But even if they have had testing at the time of diagnosis, that we're suggesting they get retested with both DNA-based NGS and RNA sequencing to identify fusions. And we hope to be able to use these recommendations to publish them and to get buy-in from some of our societies to support increased utility and reimbursement. I'll skip over the lab regulation of lab tests, but this is another obstacle that we have to think about a lot, and that sometimes gets in our way, but we persevere again. So there are a lot of challenges and opportunities in this space, and it's really great to have a big team of people who help us to think about all of the different angles. So we really need to make use of everybody's expertise and advocacy. There's a lot of opportunities for more education, more guidance, and it's important to support all of our clinical colleagues in addition to our patients and families as we're going through this process. All right, so thank you so much to everybody here and for all of my collaborators and funders. Thanks.
Good afternoon, everyone, and we are just getting started on this exciting session, and Alanna set it off at a great start with Terry Fox as a fellow Canadian and born well before Terry Fox. I watched his journey, and to this day, it's an awe-inspiring journey for me. And I also want to thank the organization, MIB Agents, everyone here for all you do to support this community, and Christina and all the organizers for just a tremendous opportunity to participate in such a special event. I was privileged to be part of the very first one in 2017, and it's a tremendous honor to be back. So with that, I have the opportunity to give you an overview of sort of what I would call the black box of moving from the lab to the clinic, and to give you a bit of an overview of the different ways that we can do this. And why is this important? Why do we, not only as a community, have to collaborate, but why do we have to collaborate with other institutions, with industry, with federal agencies? It's because we actually, in pediatric cancer and in sarcomas in general, we have a very small patient population. We're competing against breast cancer, lung cancer, colon cancer. We have to unite and work together to move into the clinic in an efficient way. We have to build on the biology. The amazing work that all of you saw the last couple days really allows us to further stratify these patient populations, further take the 600 osteosarcoma patients that are diagnosed a year in the US, but that takes it down to even smaller, what we call ultra-rare populations. And that puts even more pressure on how we collaborate to do these clinical trials. We need to build the correlative science into our clinical trials so that we continue to learn, especially as you just heard from Dr. Church. This needs to be part of how we build into our trials a greater understanding of the biology to continue to move the bar. And that requires support for correlative biology studies. And I'll show you that that's typically pieced together. We have to maximize efficiency. I'm not going to talk about trial design, but certainly the conduct. And the more we can collaborate, the more efficient that becomes. And we need to leverage typically multiple financial resources, and I'll talk to you about the 3 buckets that I put trials into. The types of trial collaborations, the first one, and this is very US, actually I should say North America centric, because Canada is involved as well, as there are also some sites in NCI-sponsored trials through Australia. NCI, through federal dollars, utilizes the Clinical Trials Evaluation Program, also known as CTEP, to support and conduct clinical trials under what are called CRADA agreements, which are Cooperative Research and Development Agreements. There's a link to the website there, which lists the current 67 CRADA agreements that exist between the NCI and pharmaceutical or biotechnology companies to work collaboratively with the government through the CTEP program to sponsor anywhere from purely preclinical studies through very selected clinical trials to what are called more open CRADA agreements, which allow for more flexibility of trials to be supported. These trials are supported through what's called the Cooperative Group Trial Network, which includes the Children's Oncology Group, the Pediatric Early Phase Clinical Trials Network, the adult groups, Alliance, ECOG, Akron, SWOG, et cetera. And this is very important for us in pediatric oncology as well as in sarcomas, because a lot, and a special emphasis, particularly in ECOG Akron, is in rare tumors and sarcomas where they have dropped the age of eligibility down into the adolescent young adult age range, increasing our opportunity to do trials in this space. In these trials, the principal investigator is appointed by the cooperative group. The cooperative group writes the trial and is responsible for the trial. But NCI CTEP holds the IND, does all the communication with the company, and all the communication with the FDA. And these are purely agreements between the company and NCI. The most common way of doing trials is here in the middle, called hybrid trials, broadly noted as investigator-initiated trials. These can take a variety of types of flavors of agreements and collaborations. Trials still under CRADA agreements and in cooperation with the NCI can be done outside of a CRADA agreement, still utilizing NCI resources. And this, again, is agreement between the NCI cooperative group and the drug company. So it would be an agreement that is contractual between COG, ECOG Akron, and the drug company to conduct the study, but utilizing NCI resources. These hybrid investigator-initiated trials can also be institution-specific or other consortia-specific. So we have heard about other types of consortia or multi-institution collaboratives that can enter into these agreements and also add in additional support from philanthropic and institutional funding. There are purely academic trials, and these are very important, especially in the cell therapy and immunotherapy space, because they leverage typically federal grants, institutional grants, or philanthropic funding to conduct typically single institution or selected institution trials. In this model, the PI and consortia propose the study and write the trial, and the individual institution or company hold the IND. The last box is the industry-sponsored trials. These are typically what you think of as a pharmaceutical trial. They are fully sponsored by a pharmaceutical company or biotech company. They can enter into direct contracts for fully sponsored studies with a cooperative group, such as the COG, ECOG, Akron, SWOG, et cetera. They can also be institutional or consortia contracts with specific companies. In this instance, the company is responsible for the trial, all aspects of the conduct of the trial, the data, the database, monitoring, and holding the IND. So what are advantages to these models? The NCI-sponsored trials, it is a single contract between the NCI and the drug company. It allows the cooperative groups to utilize a single central IRB, so a single review process. All of the cooperative groups, we don't know what happened, all the cooperative groups utilize templated protocols and consents, which increases efficiency. And the really big advantage to the pharmaceutical industry partner is that what they have to supply under the CRADA typically is the minimum requirement is drug supply, and then NCI labels and distributes the drug on their behalf. Hybrid models, big advantages. It's the most flexible arrangement. You can have multiple contracts, multiple sources of funding under these agreements, and the control of the trial design is really with the PI and study team. NCI trials with cooperative groups and the drug company, it is a single site contract for a single contract for a multi-site trial, which is of a real advantage in negotiating these. And NCI Central IRB is allowed if using the NCI resources. And there is definitely potential for adding in additional funding from other sources. The advantages of industry-sponsored trials, this is by far the most flexible for international trials. The other two types of collaborations are, there's a lot of challenges with doing broad country trials. This actually does provide typically the most financial support for sites participating in the study. And it allows, again, under any cooperative group agreement with a drug company, a single contract to deliver multiple sites. And it has the potential to shorten timelines because it has less review processes than the other two that would potentially utilize federal resources. So what are some of the disadvantages? Again, there's no perfect solution. Every type of situation, you need to weigh the pros and cons. In some of the disadvantages, the real direct communication in the NCI-supported trials is the direct communication with the company through the NCI. So if you are part of the cooperative group mechanisms, you're not necessarily always at the table for all of the conversations. It does require a multi-step review process through the NCI that goes from a letter of intent to a concept to full protocol review, as well as central IRB review. And there is limited funding to sites, and there is no funding for collaborative studies through the cooperative group mechanisms. Those have to be provided through other mechanisms. The hybrid trials, again, if we're utilizing and leveraging NCI resources through the cooperative group mechanisms, it still has to go through a multi-step review. Funding may be insufficient from a single source, and so this typically requires additional contracts for moving particularly the collaborative agreements forward, or the collaborative studies forward. Industry, again, these are industry-owned studies, and so the input of the PI or disease-specific individuals can be variable. And this is the largest burden to cite because these typically have the most requirements for data collection. And there's typically more limited access, I mean broadly, to sites because there's site selection for these studies. So in the next 3 talks, you're going to hear examples of studies conducted under these models. The first one by Dr. Reed will highlight an industry fully funded study in collaboration with the cooperative group, namely the Children's Oncology Group. You'll then hear from Dr. VanHeyst, an industry academia study. So this is a collaboration between an industry partner as well as academia utilizing other funding sources, grants, and institutional. And finally, you'll hear from Dr. Ligon, who has really, and this epitomizes the bringing up from the cell therapy, immunotherapy, a purely developed academia trial out of the labs at the University of Florida and translating that to the clinic in an academic setting. And so I look forward to these talks and to the discussion that follows.
Perfect setup. Thank you so much. I'm Damon Reed from Memorial Sloan Kettering, and here and excited to present the primary results of the open label phase 2 study. This was a screening study of activity for maintenance therapy with OST 31-164 after resection of recurrent osteosarcoma. Has two names because it is that industry and Children's Oncology Group partnership. And in the Children's Oncology Group, we called this AOST 2121. My history with this agent goes back about 9 years. When I first met at ASCO, I was excited about the agent. I'm going to tell you about the agent and the data that led to the trial, tell you about the trial design, and then we're going to talk about the primary outcomes during the next 9 minutes. And so we wrote a study, it faced some hurdles, and then we're able to launch the study in 2021 because of the industry sponsorship. And so we did go with the industry-sponsored model with OS therapies. And so the story starts at University of Pennsylvania with Dr. Patterson, who discovered this Listeria. Listeria is a bacteria that lives in ice cream, and you don't really want to encounter it, especially not if you're pregnant. But this Listeria is modified in a way that it's less virulent, it's less likely to cause actual illness. And instead of those genes that Listeria uses to make us sick, instead the Listeria lysine and LLO, a certain gene that puts pores in our cells, has epitopes, small parts of HER2 fused to it. And so the idea is Listeria goes into the host, the patient. It stimulates a broad immune response. Betsy was talking about Dr. Young was talking about CD4 and CD8 T cells, but it engages multiple aspects of the immune system, T cells included. And then along the way is also eliciting a robust activity against these HER2 epitopes. And at the time of the study design, these were thought to be rather important in osteosarcoma. So after Dr. Patterson invented this technology, Dr. Mason, an excellent veterinary oncologist, used this in a dog study. She treated 18 dogs with this agent. The dogs got what they normally get, which is carboplatin, surgery, and then got 3 doses of this bacteria. And then I am, just to show that the slides were made by both OS Therapies and myself, I would never put a picture of myself on these slides. And I was the COG partner, and so I did not put that part on. And so the canine-informed trial, they showed us these curves early on, engaging us clinical researchers with humans, and the black line was shown to us, and we said, what is the natural history for dogs? And the dotted line was 18, so they did the study. Every single dog got this Listeria, got OST 31-164, and they went back into their coffers historically and had breed matched and similar dogs that they found, and this was the outcome, the red line. And so there was a clear separation of these curves, much better outcome for the dogs that received 3 doses of this, and so that was the strongest back. There's no mouse studies. You didn't have to put this on cell lines because canines naturally get osteosarcoma. They're thought to really represent human data, and so this was the data that we had to write the trial. I talked on the right-hand side about how we think the Listeria engages, but this was what we had going in, and so. Just like the canine trial, we thought to design it similarly. So every patient on this open label phase 2 trial every patient got the medicine. There's no blinding, there was no placebo control, everybody would get listeria. Patients were all 12 to 39 years of age. All patients previously received chemotherapy, had a period of complete remission, relapsed in their lung only, and then they were eligible for this study if they could have surgery to remove all visible lesions in their lung. So that's what it took to get into this trial. Every patient was that. It didn't have to be the first relapse. It could have been the second or third or fourth or any number of relapses. And what I want to get across is a clinical trial is an experiment. It is so much money, so much time, so many people involved, but it's just like when you did your science fair project in 6th grade. You have a hypothesis, you set up, you write a 60-page protocol, and you say everything that you're going to study, what your controls are, and all of those things. And in this case, our control was a comparator arm. It was a historical study done about 25 years ago with inhaled GM-CSF. The idea of using that trial where patients also were resected and then received GM-CSF, a colony-stimulating factor, was a paper in 2016 that the Children's Oncology Group wrote. And the idea behind this paper was, let's set a bar for activity in phase 2, either in this kind of population, a population that was completely resected, or in patients that had measurable osteosarcoma. And let's say that these past studies that we completed that we consider negative, let's have those be the history, and then we can set targets for what we would say is active. All the patients on this study got a lot more of the agent than the dogs. They could get it up to 16 times every 3 weeks. And the primary endpoint was 12-month event-free survival, and again, comparing this to the historical control study. The co-primary endpoint was safety, and then the secondary endpoints were overall survival, which I will not be reporting today because we're still waiting more time, and exploratory endpoints of correlates were also not going to be presented today. They're still pending. So the patients and results, 41 patients enrolled. That means that they signed consent and they received a dose. 2 withdrew consent. They decided they didn't want to do this anymore. 1 allowed follow-up, and so will be included in the presentation that I give. 1 said, no, I'm done with this. You can't follow me up anymore, so we can't report that patient. So there's 40 patients that we're going to report today. The age range was 20 to 30 years of age. More than half of them were in the pediatric population, so most of the patients were under 18, 24 of them. And the adult patients were the other 17, about half female and half male. 13 patients completed all 16 doses. That 1 patient that stopped and withdrew consent along the way, but allowed follow-up, was event-free at 12 months. And so that leads to 14 out of 40 patients being event-free at 1 year, which was 35% of the patients. 1 patient did progress at that 1-year scan, and so depending on where you count from, the surgery or the first dose, the patient is either a plus or minus, so it's either 13 or 14, and for this presentation, we decided to do 14. 26 patients, the remainder of them experienced progression during that first year after their surgery. This is the curve of the event-free survival, and that blue shaded area is the range, so it feels good to say this is the exact percentage in truth, and our statisticians keep us honest. The 35% has that fuzzy around, so anything from around 20-some% to around 50% is that's where the truth lies, 95 out of 100 times. And the Children's Oncology Group curve is off to the side. It's not exactly like proportional, but again, there was a target pre-specified on the trial. So we have 35%, and on the historical control is 20%. In terms of safety, 12 out of the 41 patients, so the patient, before withdrawing consent, we could evaluate safety, experienced at least 1 serious adverse event. Serious adverse event, just for the definition of people that don't think about this every day, means that basically the patient had to go into the hospital. And so that was 12 out of the 41 patients. 7 of those were treatment-related severe adverse events. So while you're injecting listeria, a live bacteria, into someone's vein, people do have some reactions to that. Every patient got non-steroidal anti-inflammatory drugs, things like ibuprofen, and some other medicines to kind of blunt that. And but sometimes patients would get fevers, or the blood pressure would be a little bit off, and they would get admitted overnight for IV fluids. There was no grade 4 or grade 5, i.e. life-threatening serious adverse events reported throughout the conduct of the study. Some of the other ones included cytokine release syndrome, again, feeling like you have the flu, nausea, febrile neutropenia, fever with low blood counts, and then bacteremia in 2 cases. I'll describe those a little bit more. Whether to call something cytokine release syndrome or infusion-related reaction, those are kind of overlapping, but again, these kind of reactions to having bacteria into your veins. There was one exposure during pregnancy, and then one episode of rhabdomyolysis, one problem with wound closure, low blood pressure, and some other things that the disease progression and suicidal ideation were not considered related to the study agent. Rhabdomyolysis was seen in one patient. That's a breaking down of muscle cells, and we found that in urine and on a blood test. And so we alerted and made this new safety finding that was not seen with other Listeria products, known to the community, and people were looking for that. We did not see other cases. And then one patient grew the Listeria, the therapeutic agent, grew that in a blood culture after completing therapy. And so that was a known potential risk of this. Overall, thank you very much to the patients, the many patients and families that participated in the study, and to all open and enrolling sites. The vice chair of the study was Kelly Bailey, and Katie Janeway is the chair of the bone tumor committee, very involved in the study design and conduct. There was about 24 sites that participated and enrolled patients, including Dr. Albert. Thank you. And then also to Nicola Mason, Amy LeBlanc, and the other veterinary biologists that made this, veterinary oncologists that made this happen, along with our data safety committee, Leo Mascarenes, Brooke Fridley, Lars Wagner, and Joanne Lagme. And then the OS Therapies team was excellent to work with, the Children's Oncology Group, of course, operationals team, and George Clinical. Thank you.
I think I just clicked this. Okay. All right. Hi, everybody. First, I just want to start off by thanking Christina and everyone at MIB Agents for inviting me to come back and speak more about the clinical aspect of the work that we do in Alex Wong's lab. I'm excited to be here. So I think I'm doing something wrong. Okay. So my only disclosure is that I'll be talking about Medpacto's drug TEW7197, also known as Vactosertib. So we all know this, but over the last several decades, we've reached a therapeutic plateau for the treatment of patients with osteosarcoma, particularly those with relapsed, refractory, or progressive disease, and those with metastatic disease at the time of initial diagnosis. Additionally, the cytotoxic agents that we use to treat patients impart caustic and severe side effects. As mentioned earlier in talks, patients develop multi-drug resistance, and in addition, these agents are just simply not adequate to control micrometastatic disease. Immunotherapy seems like a promising new alternative to overcome the limitations of conventional treatment, and this has been recognized more consistently in other malignancies, and not so much in osteosarcoma for reasons that we've also discussed. As mentioned earlier, there's the lack of validated biomarkers for predicting response to immunotherapy, and then there's inconsistent clinical efficacy of immunotherapy in osteo due to tumor heterogeneity, immune evasion mechanisms, as well as insufficient immune infiltration. Okay. So Vactosertib is a small molecule inhibitor against type 1 TGF-beta receptor. It has been used in phase 1 and 2 clinical trials in adult malignancies, including colorectal cancer, gastric cancer, pancreatic cancer, multiple myeloma, desmoid tumors, as well as non-small cell lung cancer, just to name a few. And what's been shown about this drug in particular is that it's relatively well tolerated. It produced in these individuals transient low-grade toxicities, if any. And the other attractive thing about Vactosertib is that it's orally available. So patients do not have to have a central line. They don't have to spend several days in the hospital. They don't even have to spend several hours in clinic. I often get asked, well, why was the Wong lab interested in Vactosertib? And the reason why is because unlike other antibody-based therapies for TGF-beta inhibition. It has 10 times the potency of those other agents with significantly less toxicities, particularly with respect to cardiac toxicity. Okay, so why do we care? Like, what's my end point here? So tumor-associated macrophages are a dominant immune cell in osteosarcoma, and they secrete high levels of immunosuppressive cytokines, including TGF-beta. And the literature supports that TGF-beta does promote tumor proliferation as well as facilitates tumor immune evasion based on its effect on the innate and adaptive immune systems in patients with osteosarcoma. So in the Huang lab, we hypothesized that if we target TGF-beta in the tumor microenvironment, we could potentially reduce and/or ameliorate osteosarcoma. I've presented this preclinical data before at Factor, so I just put a slide to kind of highlight it. The major points being that Vactosertib inhibited murine osteosarcoma cell growth in vitro and in vivo, that tumor incidence as well as tumor volume in the mice treated with Vactosertib was less than those that were not, and overall survival in the mice treated with Vactosertib was better than those that were not treated with it. Additionally, Vactosertib still had some clinical efficacy in mice when given 4 weeks after tumor inoculation at the time that metastatic disease was present. And look at everybody so happy to be in Alex's lab, including Alex himself. So in collaboration with MedPacto, I designed a phase 1/2 open label, single arm, proof of concept clinical trial using Vactosertib in patients with osteosarcoma. Based on our data from the lab, MedPacto received orphan drug designation, rare pediatric drug designation, as well as FDA IND fast track approval for the use of Vactosertib in osteosarcoma. And so the study is still very much ongoing. So this is very preliminary data, but Christina encouraged me to share whatever we had. So the study is for patients with relapsed, progressive or refractory osteosarcoma, ages 12 and up. We've already completed enrolling 3 patients on each of the 3 cohorts at the adult, on the adult arm for patients 18 and up. And right now we're currently enrolling patients 12 to just prior to 18 years old. We have had 2 patients enroll on the 150 milligram POBID cohort. The drug I failed to mention before is dosed 5 days a week, Monday through Friday, twice daily. And then our hope is when we're done with phase 1B, we'll actually be able to proceed to phase 2. I know that's like a hot question I get asked a lot, and our hope is to enroll about 42 patients. So this is just looking at individual subject data by prognostic factors for the 11 patients that have enrolled on the study. There are 4 patients that are still receiving the drug. 3 of them are adult patients, 2 of which, I can't even read that far, but have had a partial response for quite some time and have been on the drug now for well over a year. The 3rd adult had stable disease up until last week when we determined that this adult patient also had a partial response. And then out of the 2 adolescent patients enrolled on the study, 1 of them also had a partial response so far. And what unites all 4 of these patients, if you're looking at sort of the demographics and prognostic factors on the table, is that all 4 of these patients actually had very small disease burden compared to the other patients that enrolled on the trial. So because this is still in phase 1 and the primary endpoint is safety and tolerability, the next 2 slides look at the cumulated adverse events. Essentially, most of the adverse events that are related to, or that we believe is related to Vactosertib, are either grade 1 or 2 for the most part, and they include things like fatigue, anemia, nausea, vomiting, headache, fever, as well as a maculopapular rash, none of which required a dose modification. Okay, this is just looking at spider plots for the data that I just showed you on the first slide. The plot to your left, my left I guess as well, is looking at percent of tumor burden change, with the plot on the right actually looking at size in millimeters. The two lines sort of going downward on the spider plot to the left are reflective of the adolescent patient who has had a partial response, as well as the adult patient who had stable disease until just recently and now has a partial response. So based on the data that we have thus far, we know that the risk of relapse and death for patients with osteosarcoma with metastatic or relapsed disease having achieved remission is still unacceptably high. Based on our preclinical data, the current clinical data we have, as well as many of the compassionate use patients who received this before the trial was open, we actually think we have a decent rationale for the development of a phase 2 maintenance clinical trial, which we are working on developing now and hope to open next year. I'd like to just, before I get flag shamed, acknowledge Alex, who believed in me when I was just a fellow to take on translational work in his lab, as well as Jay and Sung-hee and Suzanne, who I do these preclinical experiments with. Of course, MIB agents, to echo the sentiments of everybody else here. This is one of the most inspirational conferences to be a part of. I feel really honored to be here. And I'd like to thank the Family Fund, because of Charlotte, for awarding me an Outsmarting Osteosarcoma grant in 2022 that helped to pioneer a lot of the preclinical data. Of course, MedPacto, who is so willing to collaborate with us, and Sarah Kim, who came from MedPacto here, and all of our friends back in South Korea, as well as CureSearch and Cure Childhood Cancer for funding additional TGF-beta studies in the lab. And the last thing I just want to say is that we are the only site open in the United States right now. The other sites are opened in Korea. So if anybody wants to talk further about the trial, I'm very receptive to email. Please feel free to email me or our clinical trials unit. And I hope to come back in the future with more data. So thank you.
All right, thank you guys again for the opportunity to come and share some of the work that we're doing at the University of Florida, and thank you for the support that we've received from MIB agents. I've told Christina this before in the past that the Outsmarting Osteosarcoma grant that I received in 2022 was my very first grant, and it has really helped propel some of the work that we've done forward and now into a clinical trial. So I really appreciate the support. And I also think that we have a good group here in this panel, because we have Dr. Reed, who told us about a completed trial, Dr. VanHeyst, who's very kind of in the middle and was able to present some preliminary data, and I'm able to come here right at the beginning of our trial, where we literally just opened in March. So I'm going to be able to tell you kind of about the design of our trial, but maybe I'll come back in the future and I'll have some exciting preliminary results to share as well. So obviously I'm discussing an investigational agent that's in phase 1 clinical trials. And so I think we talked about this in the last session, but there are a lot of challenges in pediatric cancer immunotherapy. There's the diversity of tumors, there's antigenic heterogeneity, there's the low tumor mutational burden in the patients with osteosarcoma, and then there's the incredibly immunosuppressive tumor microenvironment that we have to overcome. And I think particularly for osteosarcoma, there has traditionally been a understanding that, osteosarcoma is an immune desert. You can't, use immunotherapy in osteosarcoma because there's no immune cells present. I think there are several studies, including work that I did during my fellowship, and then work that's been done by people like Dr. McEachran at the NCI, who have shown that, especially when you're looking at the osteosarcoma pulmonary metastases, these may actually be more of an immune-excluded tumor microenvironment. So if you look at that kind of edge that's outlined in green on this slide, between the tumor and the normal lung parenchyma, you do actually see immune cells and other immune markers present there. They're just kind of stuck at the edge because the tumor notices these invading immune cells, and they kind of put up a shield to keep all these immune cells at bay. And through other work that we've presented in the past, we think that it's likely largely due to these immunosuppressive myeloid cells that kind of pervade the entire tumor. And so whatever immune-based therapy we want to use, if it's going to be effective, needs to address this underlying immunosuppressive myeloid population. So what we've been trying to use at the University of Florida is these RNA lipid nanoparticle aggregates to train the body to recognize cancer. And this is work that really has been going on from my mentors, Dr. Sayer and Dr. Mitchell, for over 10 years now, before the pandemic. And you can take really any pediatric tumor, in this case osteosarcoma, take the tumor building blocks, which is the RNA, and then attach it to this custom lipid nanoparticle, which we then give intravenously. And then it really does two things. One, it goes to the tumor, and it converts those immunosuppressive myeloid cells into more anti-tumor myeloid cells. And also it goes basically everywhere else in the body that has immune organs. So like the spleen, lymph nodes, the lung, which we know is very important for osteosarcoma. And these nanoparticles get into these master immune cells called dendritic cells, or antigen-presenting cells. They're then able to take that RNA, present it to the T cell army, tell it to understand that, hey, if you see something that looks like this, it's dangerous, you need to go and find it and destroy it. And then those T cells kind of go throughout the body to go and attack the tumor. To talk a little bit about what is unique about our lipid nanoparticle, because I think especially after the pandemic, a lot of people are hearing about mRNA cancer vaccines. You can actually go all the way back to like 20 years ago. People were trying to use mRNA to target cancer with a free RNA using this protamine mRNA complex. Those clinical trials didn't really go anywhere. The second one on this screen is this lipid nanoparticle that basically is the COVID vaccine. And then the third one that you see on this slide is this anionic lipid or RNA lipoplex, which is being used by other pharmaceutical companies to target adult kinds of cancers. And what we're using in our clinical trials is this multilamellar RNA lipid nanoparticle, with the advantage here being really that it kind of has this multilamellar structure that allows you to increase the amount of RNA payload that we're giving in our treatment. And the reason that we think that this is important is because I think the COVID vaccine has the advantage that you're getting to use the most immunogenic antigen that is available to man, the COVID spike protein, which immediately gets a huge immunologic response. The problem with cancer, and particularly pediatric cancer like osteosarcoma, is that these cancers have developed in a way that they evade the immune system. So we have to do something to teach the immune system that, hey, if you see this RNA, this is dangerous. And so what we've done is by kind of making this onion skin, like alternating layers nanoparticle, we're able to increase the amount of RNA that we're giving. And if you look at this under an electron micrograph, it actually kind of looks like a virus. And so this winds up producing a profound immune response when you give this to patients. Some preclinical data that I've shown before, but essentially in different models of osteosarcoma, we're able to show that using these tumor mRNA lipid nanoparticles can cure different models of murine osteosarcoma. And we're able to show also that when you then look at those tumors, the lungs of these mice that are treated, the mice that get the tumor mRNA lipid nanoparticles, they wind up having increased central memory T cells present in the lungs. And then interestingly, and this is important for the way that we designed our trial, you can actually see that an irrelevant RNA that's given in these nanoparticles can wind up reducing the amount of tumor-associated macrophages. So an irrelevant mRNA can lead to remodeling of the tumor microenvironment, but you really do need something that is patient-specific to get the adaptive immune response. Briefly, this has entered human clinical trials for adults with glioblastoma, which is a type of brain cancer. I'm not going to go through all of these different graphs, but essentially looking at different cytokines and chemokines, you can see that you wind up seeing significant upregulation of these cytokines and chemokines within 2 to 6 hours. And this really tracks with what we see with these patients is you give the lipid nanoparticle, and within 2-3 hours, they start having fevers, chills, sometimes hypotension, sometimes neurologic symptoms in these patients with brain tumors. And then within 12 to 24 hours, those symptoms resolve. So definitely a biologically active agent. And so based on our preliminary findings in adults with having no dose-limiting toxicities up to this point, and based on our preclinical data in osteosarcoma, we're now moving into pediatric clinical trials in a trial called RNA PRIME, which is RNA lipid particles targeting pediatric recurrent intracranial malignancies and other systemic solid tumors, which I know is a mouthful. It's really intended so that way eventually we can actually add other pediatric cancer types to this trial as well. But our first 2 cohorts are including osteosarcoma and relapsed pediatric high-grade glioma. So again, I mentioned we just opened this trial in March and treated our first patient. But we have several different trial schemas that I'll try to walk us through. We have the first arm is for patients with unilateral pulmonary metastatic osteosarcoma. You do a pulmonary metastectomy, use that tumor to create your personalized lipid nanoparticle. While you're making the personalized lipid nanoparticle, which takes about 6 to 8 weeks, you use an irrelevant lipid nanoparticle targeting PP65, which is a CMV peptide, to hopefully cause tumor microenvironment remodeling. Similarly, for patients with bilateral pulmonary metastatic osteosarcoma, you can do a staged thoracotomy, and between the 2 thoracotomies, you give the irrelevant mRNA. And then for patients with unresectable osteosarcoma, you can get the tumor mRNA with a biopsy. For the 2 arms that have pulmonary metastatic resectable osteosarcoma, the efficacy endpoint is going to be 12-month event-free survival. And for the patients with unresectable osteosarcoma, the efficacy endpoint will be 4-month progression-free survival against historical control. And initially, this will be a dose escalation trial. So during the phase 1 portion of this trial, obviously the primary endpoint is safety, efficacy, and manufacturing feasibility. There are also a number of exploratory endpoints that I won't go into here, but I will highlight that in addition to getting peripheral blood, the fact that several of these arms have a second surgery allows us to potentially look at how our treatment is remodeling the tumor microenvironment. And then these are the key inclusion criteria. What I'll just briefly mention is that we do go up to age 39, and we do have count criteria and organ function criteria, with the bone marrow criteria being relevant given that we see significant shifts in the immune, in the blood counts of our patients. And then I did want to briefly mention that we are now using some of the money that we've gotten from MIB agents to go back to some of our colleagues at the vet school to combine our RNA lipid nanoparticles with radiation and a canine immune checkpoint inhibitor. And for the purposes of time, I won't go into all of the details here, but I will say that we're seeing some exciting preliminary data, so that way in the future we'll be able to iterate our clinical trial and potentially add treatment with a checkpoint inhibitor or treatment with radiotherapy and things like that. And with that, again, thank you to MIB agents and to all of the other groups that have funded our research, and for everyone who's been involved in this work.
Thanks so much. We'll go ahead and start the sort of open discussion and questions for our panel.
Hey everybody, that was terrific. I don't know if that was the best, but it was fantastic. Thank you all, especially for those of us who don't do clinical trials. That was quite an education. My question's for Dr. VanHeyst. So, unless I'm mistaken, the TGF-beta receptor functions through the SMAD proteins, which is the same pathway that the bone morphogenetic proteins use. So do we know that the drug is specifically targeting the TGF response elements, or is there overlap with other things like various members of the BMP family? And if so, is that such a bad thing? It might be a really good thing. What are your thoughts?
Hello. Okay, sorry. So we're still doing preclinical work in the lab, kind of hashing that out, and we don't have our exploratory or correlative studies back yet for the trial, because we're sort of batching those. So I'm not sure that I can give you a set answer at this point in time, with respect to this patient population, but hopefully the next time I come back, I will.
Got a couple of slides. Oh, go ahead, Brian.
Yeah, thanks, Brian Ladle. Damon, maybe a question about this trial, so the Listeria trial. So I just pulled up the protocol and looked. I couldn't quite figure out, do you know what change it was powered to see, for instance? And I would feel maybe this trial might be one of the first to kind of test this paradigm of, we just have to see better than 20%. So what is better? How much are we excited about? And then one last question as far as correlative data, right? Do we have anything as far as HER2 status of the tumors or responders? But
Keep that microphone because I might forget all three questions. I'll do the second one first. So, so the 2016 publication from the Children's Oncology Group, we were swinging for home runs with the idea that if with a really strong signal, we could bring an agent from this phase 2 design, from either the adjuvant setting, after surgery, or with measurable disease, bring it to the front line, add it to MAP to get enough power to have a positive study in a population. So on this study specifically, we had two thresholds. One was with the 39 patients treated, we ended up having 41 patients treated. Anything under 30% at 12 months was considered ineffective, move on, nothing to see here. Anything over 50% was considered positive, this warrants further investigation. We never defined on purpose, never defined 31 to 49%. In talking with Dr. Pettit, the chief science officer, he said when they discussed this internally, it's effectively inconclusive for the SAP, the statistical plan, and based on the stats, there's a positive trend, but more data needed to confirm. So that was his view in communication yesterday. In terms of HER2, when the dog study was done, there was not a clear correlation with HER2 expression. There was a second dog study done, what's called a phase 3 with 118 dogs, published in May. I read that on the flight here. It was fantastic with really rich correlates. That was an unselected trial because of the finding that HER2 and the relationship seemed unchanged from the 18. So we elected here as well to not require any kind of HER2 threshold. In terms of looking at HER2 or any isoforms of HER2, those are all planned in the protocol, but no data yet, but we did collect some blood. What was the last question?
I just thought of another one. No, if you recall the Dana-Tuximab study, there was maybe some thought about how many relapses patients had. Was there any sense at all in this trial, like first relapse patients or multiple?
Yeah, I would say, as we look back, any sense for Dana-Tuximab of what? Oh, multiple relapse. Okay, good. So, this 2016 paper and having a bar, like if you can get 50% of people at EFS of 12 months, has spawned lots of osteosarcoma trials, which is good. And I was part of 4 of them, and it's been great for research. I think that people now can just at any site, like pop up a study and say, this is a universally kind of standard bar for activity. So that's been great. Unfortunately, I would say unfortunately, there are patients waiting for all these studies. And like we always say, it's going to take this long to accrue a study, and it always accrues way faster. And so osteosarcoma in the relapse setting remains an unmet need. But you mentioned dinutuximab had a very similar study design, also landed around this 30-some% and stuff. And so that was your third question. The first time was how much more than 20%? And I think we're struggling with that right now. So I think if you try to hit a home run and try to take a bar and go 2.5 times higher, we've not seen that yet, but that's a huge bar. What do you do when you see something? We don't know yet.
Okay, I have a question for Damon Reed. So regarding OST 31164, two questions. When will this be ready? And then the other question is, who is this going to be for in the future? For example, will this only be for patients that have recurrence of osteosarcoma, or would this also be used, for example, to prevent a recurrence for a patient that finished MAP, NED, high risk, doesn't want a recurrence? I guess, who do you envision this for?
So the first question is, when will this be ready? So this was a phase 2 study. So for everyone, a phase 2 study to me is screening for a signal of activity. So let's say this was a far more common cancer, breast cancer, something much more common. A phase 2 study is like, how big of a difference do we think this is from the others so that we could design the real study, the confirmatory phase 3 study where we show without a doubt that it has activity. And so right now we have what I presented that compared to this one study from a long time ago, Dr. Ladle just mentioned another study that's been more recent that had a similar finding. So now this is what we think the activity could be, but now it would have to go, some further tests would have to be more definitive towards regulatory approval. So this will be ready after the FDA and the sponsor, OS Therapies, works out a plan to say what would be enough to get a regulatory approval to make this drug available. I imagine that it would continue to be explored in this setting, in the minimal residual disease setting after surgery. That also answers one of the Slido questions, which would be with resected bone metastases. And so that complicates the study design because those patients do less favorably, so you start to lose you're trying to compare apples to apples as much as possible and minimize the variability. Did I answer the questions? But it's not ready yet. I mean, it won't, I don't.
Right, it's not ready yet. But do you think in the future you could envision it being used for patients that are NED to prevent a recurrence?
I think that's what it would be studied in next, yes.
Thank you.
Damon, one follow-up question. Sort of thinking about, we've talked a lot about cross-species comparisons and how the dog trial led to this. The paper you mentioned in the phase 3 in the dogs was not a positive signal, and I'm kind of curious both if you have thoughts as to why there's the difference, and had that trial come out first, would you still have wanted to run this trial, or would it have potentially led us to miss this positive result?
Yeah, I mean, a fair question. So this was based on 18 dogs, but it's a whole wormhole to go down, which would be what is the preclinical threshold for a positive study. And 18 dogs was far better to me than 5 mice and far better than 3 cell lines. And so it was, and it was exciting data, and the canine community was very excited about this. And it is unfortunate that amidst about middle way through this study, the larger dog study was first presented to our team as negative. I mean, those big changes on the EFS curves were not seen at all. I mean, and so that study was 118 dogs, all receiving the medicine, and then compared to a previous study that they did. And it was prospectively planned that way, so it's effectively a randomized trial, but done a little bit different than we would call that in a human trial, but still a legit trial. It's a tough question. If there was more robust data that there was no signal of activity in the canines, would we have run this study? I don't know that it would be a question more for the sponsor. I think that we're always kind of looking for agents with some signal of activity. But usually when you do a phase 2 and it's not confirmed in the phase 3, your enthusiasm would go down a lot. It's a fair question.
To follow up on the question, the previous question or two ago, what about patients that are coming out of treatment and if they had circulating tumor DNA in their blood, is that something that you would consider running the trial for? And how much further would that field have to evolve for you to say that's equivalent to scans and that's worth doing the trial on?
I'll go back to when I described a clinical trial as a scientific experiment. I think you guys are asking really good questions, but you're changing a variable. And I think when you change a variable, you kind of start all over again, unfortunately. Someone said yesterday that a clinical trial is not exactly something along the lines that's not a very good tool or something like that. But it is difficult. I always think now when I design a trial, is this going to be still exciting 7 years later? Because that's when you're going to be presenting the results, right? And it's complicated. So what we know about osteosarcoma you've seen over the last couple days, you've seen the movies and single cell data, and no one thinks it's one cancer anymore. It's a whole bunch of different cancers altogether. Like there's all these different clones. When we started this study, we thought osteosarcoma is the same cell over and over again. Cut out a piece, grind it up, do some sequencing, and that's the cancer. There's no variability inside. It's a clone. It's just this one cell kind of going out of control. So now if we would change the status not from surgery, but surgery and ctDNA positive, I would imagine those patients would have a much higher risk of relapse. So I would imagine if we just change that one variable, the EFS of 20% would be even harder to achieve. You're probably you're making the agent have a harder hill to climb. So the answer, Natalie has taught me, the answer is randomization. Whatever you do, if you're going to change a variable, then at the same time as you're studying your study, randomize patients to something else and compare. And that kind of gets rid of a lot of the problems of changing a variable. Yeah?
Maybe for Dr. Weigel. So I've heard, right, I'm not yet a senior person, right, I'm not a terrible young person either, but I've heard so many stories. I sit in the room with the disease experts, and we have come up with this trial, and then you hear they said it wouldn't be a good trial, right? Like something was terribly flawed, and I realize that resources aren't unlimited, but for instance, like the NCI-sponsored trials, is there a way to maybe modernize that process at all, or is there things under, in the works there where I do feel like it's sometimes obstructionist at some of those levels to proceed with clinical trials that I think have really, really intriguing data. So.
Yeah, so I really appreciate your question, and I think it's complex because I do think you're absolutely correct. In the cooperative group setting, if it's a trial being done under that arrangement through the CRADA mechanism with NCI-CTEP, the initial step to go into that sort of depends a little bit whether it's a phase 1 or phase 2 or phase 3 trial, but suffice it to say that the first step is essentially what we call a concept. So it's not really a trial, it's the expert team putting that idea forward to the review process at NCI-CTEP once it comes out of the cooperative group network. There's a couple steps in that initial concept review. There are what are called disease-specific review teams at NCI-CTEP that are also filled with other experts not conflicted that also review it. And then it's also reviewed by representatives of the company so that it aligns with their development plans and what they've agreed to under the CRADA. So some of it is in that initial step is does it fit with that actual alignment, both with the company and with NCI-CTEP. And then typically it's an iterative process. I would say it's not very common that at least in my experience, that something is completely shut down, but it may not be exactly as the experts in the disease-specific committees for the cooperative groups had envisioned. And sometimes it is a bit of give and take to marry all those all those agendas, if you will. And I think the biggest thing we can do as a community and as people who do see ourselves as championing the ideas of the disease expertise is to continue to try to modify that. And I think one of the big things, I'll use the example Damon just cited with regards to using data to push back. So one of the things in osteosarcoma was to push back with data for this comparative group based on multiple studies done within the cooperative group to say this is what we see as meaningful, and then try to educate the community. So I think some of the onus is also on us to say, let's put the data together to try to move the bar and move that forward. I also think that as much as we can try to educate colleagues within industry helps us as well. Thank you for your question.
Is it on? Yeah, okay.
I think we have time for one more. We're a little bit over time.
Do I
but
get the last word? Excellent.
Yeah.
All right, so I was involved in the canine confirmatory trial of the Listeria HER2 product, so I just want to clarify a couple things. You can pull this paper, and please do read it. There's a couple of really important things about it. So the dogs only got 3 doses. So humans got 16, up to 16. So one of the things that we pretty quickly realized is that if we were going to do this all over again, we would give them more doses of the product. Because dogs, if anyone's not familiar, they get amputated, they get carboplatin chemotherapy, and then they got this product sequentially. So there's a couple things we could have done differently and might do differently in the future to pursue it. So just so everyone's on the same page about how it's administered. The second thing is there's absolutely a signal of efficacy in some of those dogs, and it has everything to do with the way their immune systems respond. So that paper's got really lovely correlates for immune response like fever and cytokines and cellular changes that really support the notion that there's a subset of dogs that respond and will do better with that than without it. I guess the final thing is that we've devised a transcriptional signature in dogs and applied it to humans, and it predicts outcome. And that transcriptional signature divides patients into 3 groups: immune-enriched, immune-depleted, and immune-enriched with an ECM component of vascular and fibrotic proliferation in there, which we think might act as this exclusionary barrier to immune cells trafficking into the tumor mass. And the immune-enriched patients, both canine and human, are the minority. And we're talking about like 200 cases per, across dogs and similar-sized human transcriptional data sets that have clinical outcome data that we could pull from the public domain. So I think any form of immunotherapy has to take the first patient sample and their immune TME into account, because you can't really expect anybody's tumor that is wholly immune depleted to have a response to an immunotherapy. That's just how I'm thinking about it.
Thanks, really important points and for us to think about in our human immunotherapy studies as well. Thanks everybody for a great session and a great conference.